Welcome and welcome back. Before the break, we met Paul Cadol, who talked about soil ecology in relation to climate change. Now we're going to learn more about the molecular mechanisms that control plant development. So please welcome Markus Schmidt.
Being a researcher can be extremely frustrating because normally things don't work as we expected, so we have to constantly re-evaluate our hypothesis. However, when things start to work and we start to understand why a plant responds to an environmental stimulus as it does, that I find extremely rewarding. My name is Markus Schmidt and I'm professor in molecular plant cell biology at SLU since March 2023. Plants, in contrast to animals, have the ability to generate new organs throughout their entire life. And this enables plants to adjust their growth and development to their surroundings. However, the capacity to do so is challenged by climate crisis, global warming. And in my lab, we want to understand the molecular mechanism which enable plants to adjust their growth and development to their environment. We are particularly interested in a molecular process referred to as alternative RNA splicing, which enables plants to make better use of their genes. We study this in the context of the regulation of flowering time, which is basically how plants decide when it is time to flower. And this has a direct consequence on crop yield in many plants. Our research is mostly concerned with basic molecular biology, so there are no immediate applications. However, we hope that our results will ultimately contribute to the breeding of plants which are better equipped to deal with the consequences of global warming. Personally, I believe that basic curiosity-driven science is the key to tackle those challenges and solve them for future generations.
Welcome, Markus.
Thank you.
So I understand you have discovered a plant that responds strongly to changes in temperature.
Yes.
And the plant can change its development through changes in the protein production instead of changing its genes.
Precisely.
Mhm.
Yeah.
And now you're trying to uncover the mechanisms that make it so sensitive to its environment.
Exactly. Yeah. That's right.
My question is, how do you know which plants to study to solve the puzzle? How do you find the mutants that respond differently?
Yes. In principle, as plant biologist or as any biologist, we have two options to find mutants. One is just by pure chance, looking in the field, which plants look different, and then try to uncover the molecular mechanism or the genes that are involved that give rise to this phenotype. More lately, with the advent of mutagenesis, targeted mutagenesis, more recently CRISPR-Cas9, it is of course possible, based on all the prior knowledge that we now have about plants, to specifically mutagenize certain genes and then study the effect on plant growth and development. So where traditionally we basically would randomly introduce mutations into the genome and then look for the plants that have the effect or show the effect that we were looking for. Now, with all the prior knowledge that we have, it is often possible to turn this around and start with, we have the genes, we know what we would expect, and now we have the possibilities to make mutants specifically in the genes and see if our hypothesis was correct.
And how do you do when you mutagenize a plant?
Well, if we want to do it targeted, the best way really is CRISPR-Cas9, because it is the gene scissors exactly that is so controversially discussed, I would say. And that is really the best way because it is relatively fast and it's relatively cheap to do.
Okay. Well, in 2019, you were appointed a Wallenberg Scholar by the Knut and Alice Wallenberg Foundation. And recently, you were given a 5-year extension of this grant for independent research. So congratulations.
Thank you.
What does this grant mean to you and your group of scientists?
Well, as I said, I mean, what we do is basic research, and that usually means that we have developed hypotheses which we need to test, which in plant biology it simply takes time. So having a 5-year program like the Wallenberg Scholar with rather generous funding really means the world to us, because it gives us the security to test some hypotheses and also maybe sometimes go out on a limb and try some crazy ideas that we had that normally we as researchers, when we are starved for money, might not be able to do. So it gives us a lot of flexibility and freedom to try stuff. And that is sometimes a little bit limiting in this system.
I can understand that. So now the stage is yours for your presentation. So
Thank you.
please. And I would like to introduce Markus Schmidt, professor of molecular cell biology with specialization in plants, talking about regulation of plant development by the environment.
And why we should care.
Yeah,
Exactly.
Yeah.
So I added this little subtitle with a nod towards the sustainability goals that already were mentioned in some of the presentations today by the UN. And of course, sustainability is one of the, let's call it, trademarks of SLU as a university. And we have already heard from Stephanie, those of you who attended her presentation, about some basic aspects of plant biology. And I would just like to remind you, and although you on the follow this lecture on the live stream, that plants are sessile organisms. And that has a serious consequence for them because they cannot evade adverse conditions. They have to deal with them, whatever nature throws at them. Be it drought, be it flooding, they have to deal with it. And one way to do this is to adjust their growth and development accordingly. And they can do that because, different to animals, plants have the ability to constantly produce new organs. They produce leaves, they produce shoots, eventually flowers, and of course, underground roots. And that gives them a lot of flexibility. And they need it really to adjust to their environment. I mean, here's a picture shown from some forest up in Umeå, where I was for the past 8 years. And I mean, this is a really challenging environment. It's high light intensities in February, March, but it's real freezing cold. So how do plants really cope with these diverse environments? But even if you think about more benign environments, such as this crop here, this field, I mean, the plants still are facing changes, daily fluctuations in light intensities, changes in temperature, attacks by pathogens, water availability. So plants need to constantly adjust their growth and development to these environments. And by and large, they do a fantastic job about it, because otherwise there wouldn't be so many plants, and they are really everywhere on this planet almost. And unfortunately, this ability to adjust is not limitless. There are potential limits to this, and these limits are being challenged. And we're going to see pictures like this probably more frequently in the future because of increases in temperature due to climate change. And where we will end up in 50 or 100 years at an average global increase in temperature of 1.5, 3, 4, or 4 or 5 degrees, well, that's up to us and our behavior. We as individuals might think we cannot do much, but we can at least make an effort. But what this means in terms of agriculture and forestry is that in many parts of the globe, of the world, the yields of crops and other bioproducts are expected to go down dramatically. It might not be so bad in, let's say, Canada or parts of Scandinavia, where the overall increase in yield might actually be happening. But overall, globally, this is quite a challenge. And it also happens or will occur mostly in countries which have a large population growth. And that will also continue for the next 50 to 100 years. So and if you add this and form this simple equation, then what it means is that eventually we will have to grow more food for more people on less arable land. And that is really a challenge for plant breeders, because what it means is that we have to make the plants more efficient. We have to increase yield. And probably to an extent similar or even exceeding what was happening during the Green Revolution in the 1950s and 60s. And afterwards, of course. So where does my research group then come into this? Well, I have a confession to make. We don't really work on crops. We work on Arabidopsis thaliana, a model organism. And we also don't look at yield specifically. What we have been working on over the last 20 years in my group was starting with the regulation of flowering time. So when does a plant know when to flower? This is mostly governed or often governed by day length and temperature cues. And also temperature cues have other effects on plant growth and development. And we usually do not work on specific genes. We very often look at what happens in the whole organism. We look at all genes simultaneously using high throughput technologies. And all of these three subject areas over the last 20 years, about 5 years ago, culminated in triggering our interest in a process that's called alternative RNA splicing. So that is really what we have been working on as part of this 5-year Wallenberg project that Malin already mentioned, and that we will continue to work on in the next 5 years. And we will mostly focus and stay with the model organisms that we have been working on, Arabidopsis thaliana, for the very obvious reasons that I have listed here. It is small. It has a rapid life cycle of 8 to 10 weeks. We can make transgenic plants quite easily. For many of the genes in Arabidopsis, and it has around 30,000, we can just order mutants these days because people have made those for us already. And it has other advantages as well. And these 30,000 genes in Arabidopsis thaliana are, of course, encoded in the DNA. And I would just like to remind you of the information flow in a cell, which we usually say the information is encoded or stored in the DNA and eventually gives rise to a protein, a molecular machine that carries out a function in the cell. But there's no direct connection between those necessarily. There's an intermediary, and that's the RNA. That's basically a transcript that is based from the DNA. It's a copy, and this copy basically carries the instruction to the cell how to make a protein. Unfortunately, it's not quite so easy, because for many genes, the RNA needs to be processed. And that is the process of RNA splicing, among others. So we make RNA first, and we convert it in what we call a mature or messenger RNA. And what happens in this process is that the RNA, which is a transcript of the whole DNA, only encodes certain regions of these RNA molecule are really informative and important to build a protein. And these building blocks are called exons. And during the process of RNA splicing, what happens is that these exons are excised and stitched together to form the blueprint for make a protein. And this is where it gets really, really complicated, because a single gene, which forms a single RNA, can give rise to multiple messenger RNAs, which in principle can give rise to different proteins. And that's the process of alternative splicing. And what the organism gains by that is the ability to have one gene, but multiple proteins that have eventually different functions. So you become, as an organism, a little bit more flexible in how you respond by shifting the ratio between these alternative mRNAs. So this is the outcome. We have one gene, potentially multiple messenger RNAs, potentially multiple proteins. Not all of these RNAs or proteins are functional. Some of them are degraded. And here I just give an example of 3 RNAs and proteins, but this system can be extremely complicated. And I think the world champion right now is a gene in fruit fly, Drosophila melanogaster, where a single gene can encode 38,000 different protein variants. That's 3 times as many genes as the genome encodes. So it can get horribly complicated. In plants, we don't have anything like this. At least I'm not aware of any example. But we have plenty of examples where genes are alternatively spliced. And here I would like to introduce one example from our own work, published 10 years ago. And it concerns a gene called flowering locus M, or FLM. And the orange boxes here are these exons, these building blocks which give rise to the protein. And what we and others found 10 years ago is that in response to different temperatures, different of these building blocks are used. And the variant on the top, which includes the building block number 2, the exon number 2, that is preferentially made under cooler temperatures. And this protein variant that results from this messenger RNA actively prevents flowering. The plant steps on the brake when it gets colder. The other variant, which includes this exon number 3, is preferentially made under slightly warmer temperatures. And that is a non-functional protein, might even act as a dominant negative protein. And that enables plants to flower. So by switching the ratio between these transcripts, the plant has a way to fine-tune its response to temperature and regulate flowering. Now, here is an example then of how serendipity comes in, because of course this triggered our serendipity in science comes in, because this triggered our interest in RNA splicing. This is where it all started. And we were looking for the factors, the genes and proteins that would regulate this alternative splicing of this particular transcript. And I can tell you, we never found it. We looked quite hard. And the reason why we not found it is that the splicing process is really complicated. It involves hundreds of genes and proteins in the cell to decide where to exactly make these cuts and which exons to join. But what we found, and this is really work by one previous PhD student, Giovanna Capovilla, is a mutant in a gene. And normally, when we grow plants at different temperatures, this is a control plant, it pretty much looks the same at 23 and slightly lower temperature, 16 degrees. But this particular mutant was different. It was extremely temperature sensitive. And even if you don't look at plants very closely, you might appreciate that this is not how a plant should look like. So, and this kind of very temperature sensitive phenotypes was quite unusual. So that triggered our interest, and we started to look for other plants which might have similar phenotypes related to RNA splicing. So the question really was, is porcupine a unicorn? Is it a one of a kind, or is splicing in general an important process, molecular process involved in temperature responses? So this is where we started. And as I said, there's hundreds of proteins involved in the regulation of splicing. So we couldn't look at all, but we found quite a few in which these temperature responses were different, particularly at cooler temperatures. So we had our eureka moment for really just a very short moment, because we said, okay, great, temperature is important for it, or splicing is important for adjusting plant growth and development, particularly to lower temperatures. And as always, when you think you have figured it out, nature throws you a curveball, and we found this other gene, which is particularly important at warmer temperatures. It's also involved in splicing and RNA processing. And this is really where the prolongation of Wallenberg comes in, because this is what we're going to do in the next 5 years. We will try to understand at more basic underlying molecular mechanisms of RNA splicing in plants, in particular in relation to temperature. And we will also start seeing if we can employ RNA splicing as a target for breeding more resilient plants. And the hypothesis is simply that if plants do well at moderate temperatures and non-ambient temperatures, but no matter how we challenge them, splicing becomes more important, then maybe by raising the overall splicing capacity in cells, plants become more resilient. It's a naive assumption, a naive hypothesis, but we would like to test this. And with that, I'm at the end. And if you have any questions, I'm happy to take them. And if you don't have any questions now, feel free to contact me later. Thank you very much.
Thank you very much, and I will encourage you to ask your questions now in this session. Yeah. Well, you showed that a mutant of Arabidopsis thaliana, the king of model plants, according to Stephanie Robert, at least.
Or the queen.
Or yes, I think it's a she.
I think so too.
Yeah. The queen of model plants, called backtrav in Swedish, and mouse earcress in English. Yeah. Well, anyway, you showed that a
tailcress, yeah.
Okay.
Whatever.
Yeah, tailcress. You show that this mutant behaved in the same way as a non-mutated variant at 23 degrees,
Yes.
but not at 16 degrees. What is it that causes the mutant in the example to fail to adapt to a lower temperature?
Well, that is exactly what we have been trying to figure out for the past 5 years.
And what did you find?
It is very complicated because RNA splicing is a complicated process. It involves many factors, but it also affects a large number of genes. So at any moment in a given cell, there's 20,000 of genes active. Each of these make an RNA, and each of these RNAs need to be processed. So now we mutagenize one of these factors involved in splicing, and that affects the splicing of many of these transcript thousands. So while we can identify global trends about what goes wrong in the cell, like these hormones are not balanced anymore, we cannot really pinpoint it to one or two genes which say, okay, this is why these plants look so bad. And I guess on Facebook you would say it's complicated.
Yeah. And how do you cope with that, the fact that it's so complicated?
Well, you just have to accept it.
Yeah.
It sounds a little stupid maybe, but
that's why you're a professor.
because I'm stupid, yes.
No. You can cope with it. Yeah.
No, you just have to accept that it's sometimes not possible to really pinpoint the exact molecular mechanisms. I know it's RNA splicing is affected. We know certain genes which are particularly important in that process and will contribute to this phenotype. But it is still very difficult to understand what exactly is going on. But I mean, that's the beauty about science, that
of course,
there's always another question. I mean, you have find something and that leads to an hypothesis, you test this hypothesis, you reject it, or you, if you're lucky, you verify it, and then that leads to new questions. So, and that's what I find so exciting about basic science.
Yes, any questions?
Well,
you use GMO in your work,
yes,
which I met with a lot of skepticism by many. Can you elaborate on that? What do you think about the discussion about...
My personal opinion of...
Yes,
I'm happy to share that with you.
Yeah.
Because what many people don't realize, I mean, if I were to ask you now who has eaten genetically modified plants, I think many would say no, and I would never do that. But you do it all the time. Because the way genetically modified organisms are defined in Europe is that anything that originates from classic mutagenesis, and that includes mutagenesis by chemicals or radiation, are by definition GMOs. They are just exempted from regulation. That's why we can grow them in the fields without asking for permits, basically. And the big question about CRISPR-Cas9 is basically, is that a safe new way to do mutagenesis, to introduce mutations into the genome? And I personally think it is, because before we introduced thousands of mutations without knowing what we would do, which genes we would mutagenize, with CRISPR-Cas9, in principle, we can hit one gene very specifically. And I don't really see why that should be worse than treating a plant with chemicals and not knowing what the outcome would be. But that's my opinion.
Yeah. Well, thank you for that. We have a question. Well, a question over there, or two maybe. We can come back to the GMO later.
Thank you for your talk. In a broader perspective, will your research help me to get more tomatoes in my greenhouse?
I don't really know if you want to have more tomatoes in your greenhouse. I would say maybe tastier tomatoes are very often what we would desire. I mean, I have a colleague from the Netherlands here, so I shouldn't make any fun about Dutch tomatoes. But there is always this balance between how well and how big fruits grow and how much flavor and sugars they contain. My work might contribute to improving this. Because, I mean, we work on temperature responses, but in general, splicing and a better understanding of RNA splicing will affect basic cellular mechanisms. And it will also be important for engineering endogenous pathways in the cell that lead to better resilience, faster growth, more flavorful fruits, for example. So yes, but that's far down the road. At this point, we just want to understand how does it work in principle before we take the next step.
Do we have another question? No?
Yes. I
Yes.
think-
Yeah, this was very
Is a microphone?
interesting. Maybe this is a stupid question, but I'm trying to understand the connection between RNA splicing and plasticity in plants.
And what is plasticity to start with? Just
Plasticity is basically the ability of plants to adjust. So basically, an organism can have different shapes and forms depending on their environment. This interaction between the genome, what's encoded in the genes, and how the environment really shapes the
okay.
organism.
So now back
And
to past
of course,
question.
RNA splicing is a very important part of the development of plants. So I'm pretty sure that it will turn out to have an impact on phenotypic plasticity, for example, about when it comes to differences between natural variants of plants that have different capabilities to adjust to stresses, for example. I don't think that anybody has really studied this particularly in detail, because each of these processes, plasticity and RNA splicing, are very complicated. And if you add those two together, I mean, I always put it into my grant applications, but it's very difficult to work with, because it's not just adding two things, it just grows exponentially in complexity, sorry. So, but yeah, I'm pretty sure it does contribute to it, but I can't give you any good examples.
Well, we have an email question here,
Yes.
and I'll try to read. I read it as it's written here. Does splicing play a similar role in species with small as compared to larger genomes, like, for example, in fruit fly and spruce? Okay.
Yeah, that's a very, very difficult question. I mean, to answer, in general, I would say probably yes. It's not really dependent on the genome size. For a long time, we were not really sure how complex RNA splicing in plants would be. It was a concept first discovered in yeast and animals. And in general, I don't think that the overall size of the genome is really what matters. It might affect which exons are more easily accessible and can be incorporated into the final messenger RNA to form a protein. But I don't really think that there's any evidence that, for example, Arabidopsis has much less splicing than spruce, which has a 20 or 30 times bigger genome, to just stay within different plants.
Well, a last question then. Well, Stéphanie Robert, she talked about when she talked about the future development of her research. She mentioned that they will look into new model plants. What about that was one trace.
Yeah.
Will you stick to the Arabidopsis, or will you also look at
We
into
will
new
definitely
models?
stick to Arabidopsis for answering basic biological questions,
Okay.
because it has so many advantages. It's established, it's fast, so never change a winning team, I would say.
Okay.
But we also start working on poplar, for example, and we are open to work on other crop plants, for example. And that's part of the Wallenberg project, again, if I can mention that, because that's part of the stuff that I propose to do there. So yeah, testing some hypothesis how RNA splicing can be used to increase resilience.
Well, good luck then with that.
Well, we will need it.
I would like to thank you very much, Michael
Thank you,
Schmidt.
Molly.
And well, let's give him an applause. This was today's last lecture, and tomorrow we welcome 4 more professors who will talk about their research, and we will start at 9 o'clock, and you can find the program at slu.se/professorer. So welcome back, and thank you for today.